Our next talk will be by Vladimir Grouza, who is a PhD student in the Quantitative Life Sciences program at McGill University. He'll be speaking about his work on histological validation of rPCA-derived myelin water fraction imaging in a panel of variably hypomyelinating mice.
Awesome. Can you guys see my screen okay?
Yes, perfect.
Awesome. Thank you for the introduction. So like Chris said, I'm going to be showing a new myelin water imaging processing technique on a panel of hypomyelinating mice, which I'll introduce shortly. So I'm going to go over a brief introduction to myelin water imaging, even though we've seen a number of great talks on that topic today, and then introduce rPCA processing and show how we integrated that with a panel of hypomyelinating mice and how we further integrated that in the MRI G ratio framework. So basically in myelin water imaging, as we've seen before, we leverage the differential properties of T2 and T2 star relaxation to essentially split a multi-echo experiment into myelin water components and intra and extracellular components. And typically, myelin water imaging experiments are acquired with a multi-echo spin-echo sequence, like the experiments of Whittle and McKay, and later with Catherine West. And alternatively, MGRE, multi-gradient echo experiments, which are T2 star weighted, and have a couple of cool advantages for clinical applications, such as faster readout, low SAR, and physiological noise. They have more physiological noise. This is an unfortunate side property of those. But basically, people traditionally like to do this kind of calculation of splitting these signals using either non-negative least squares fitting or three-pool magnitude model fitting of white matter, which involves the sum of three exponentials using non-linear least squares fitting. And the myelin water fraction is basically just termed as the myelin water component over the sum of the total signal of the spectrum. So in contrast to this, our colleagues in South Korea developed this technique called BSS rPCA. It was developed by James Song et al. In 2020 and published in IEEE TMI. And basically, they use a series of matrix manipulations on the entire echo train to separate the MGRE signal into two what are called unit rank components. So a quickly decaying L2 component, which is attributed to myelin, and a more slower decaying component called L1, which corresponds to intra and extracellular compartments. And unlike the previous models discussed, this is completely model-free. It's computationally expedient, which means 5 to 10 minutes computation for whole brain coverage. And it also performs denoising simultaneously by sequestering any artifactual signal into what's called this sparse S component. And fortunately, we've re-implemented it in Python, and we're happy to share it. So in algebraic form, what this looks like is the minimization of a convex objective function J, such that we extract these components L1 and L2 from M in a patch-based RR manner, while also enforcing Hankelization to enforce unit rankness and therefore mono-exponentiality of the signal. And then finally, we compute myelin water fraction for the whole brain by taking the ratio of L2 at the first echo time to the sum of L1 and L2. So that's a lot to take in really quick, but pictorially, what this machine looks like in its outputs is, if we scan a mouse with a gradient echo sequence and look at our results as a function of echo time, we see that this L1 output decays much more slowly than the L2 component, and as well as we get some noise sequestered into the sparse. Fortunately, our ex vivo scans were pretty high resolution, so not a lot of stuff comes into the sparse. But if you further go ahead and perform mono-exponential fitting on each of these components as a function of echo time, you see that in the original source signal, you have a pretty consistent proton density map, but you have hypointensities along the white matter in your L1 PD map, and you basically get a white matter skeleton in your L2 map. So that's kind of reassuring as to a preliminary kind of confirmation that this machine is doing what it's supposed to be doing. And if you analyze the corresponding T2 star maps and bin them as histograms, you can see that based on the histogram distribution of T2 stars within these T2 star maps, that the L2 components are considerably shorter than the L1 component. Suggesting that something is being split here. And then finally, if you look at an individual voxel in panel C, you can see that the MGRE signal M in gray is being decomposed into this slowly decaying and rapidly decaying signal. So we tested this thing out to validate it on a very interesting panel of hypomyelinating mice that were developed by Human Bageri under the supervision of Dr. Alan Peterson here at McGill. And basically what they did was they looked at this MBP gene locus, and the MBP gene, as many of you probably know, is responsible for the transcription of myelin, right? So in fact, the shiverer mouse is basically a total ablation of the entire MBP gene. So what they did was they figured that if they selectively remove the M5, M3, or M1 transcription enhancer sites, they can get less transcription of the MBP mRNA, and thus less translation into MBP protein by oligodendrocytes, and thus less myelin elaboration. So while this isn't meant to be a model of any specific disease, we thought it'd be great for calibrating myelin water fraction measurements. So we scanned 23 animals with these graded genotypes ranging from wild type to what I'll call single, double, triple knockouts, and their corresponding MBP mRNA quantifications are listed right here. So we have the wild type, and then we have full ablation shiver, and then we also have this putative hypermyelination mouse with an exorbitance of MBP mRNA. So our imaging protocol was performed ex vivo at 7 Tesla. These are our imaging parameters, but the most critical of them are the fact that we did this at 100 micron isotropic resolution with 2 millisecond echo spacing. We corrected for gradient effects and even odd echo misalignments. And then we registered this every single map to an anatomical atlas and parcellated them. So this is kind of what our registrations look like. And these ROIs that we studied, I'll explain in a second. So 3D rendering of these ROIs reasonably well sample white matter from the most rostral to the most caudal positions. And we looked at the cerebellar white matter, internal capsule, 2 ROIs in the corpus callosum and the anterior commissure. And this is just a cool picture of what this ultra high resolution myelin water fraction map looks like in a wild type animal. So if we apply these parcellations on our entire group, our entire cohort of mice, we, through using ANOVA, in this case Tukey's HSD test, we see a statistically significant difference between wild type and these progressively hypomyelinating animals. But interestingly, no additional myelin water fraction is observed for the putatively hypermyelinating knock-in, which is interesting because this essentially suggests that there are post-translation transcriptional mechanisms that decrease the MBP product and don't participate in myelin elaboration. But more interestingly is if you actually take all these data and plot them versus their corresponding MBP mRNA, as evidenced by RT-PCR, you get a really, really nice regression plot where myelin water fraction in all ROIs studied is very robustly correlated with the MBP mRNA. So we decided to take this a step further and also look at MBP immunohistochemistry. And we basically took 5 brains because it's pretty expensive and time consuming to do this kind of experiment. And we cut the whole brain and we manually lined up a few sections from several animals, stained them with the Ventana OmniMap protocol for myelin basic protein and light chain neurofilament. I'll talk about the LNF later. And then we developed an in-house pipeline for 2D MRI to 2D IHC image registration, and we quantified the degree of staining using a deconvolution operation whereby a virtual diaminobenzidine image is deconvolved from a red, green, blue representation of a histological slide. So we took all this data together and we co-registered our histology to our 2-dimensional slices, and they're essentially all aligned to these 3 sections on the rostral caudal axis of our template. We studied the same ROIs that we studied in our 3D parcellation, and in these 3 levels, when you look at this VDAB virtual diaminobenzidine image deconvolved from the original IHC image, you see that DAB intensity and MWF intensity correspond one another quite well. And basically, if you then apply these parcellation masks to these results and normalize by the mean intensity of the wild type represented in gray, then as a function of genotype, our results cluster out quite nicely, and we get a nice correlation between the actual MBP protein and the MWF that we observe. So then we decided to do a control experiment for diffusion to control for axonal density. And the two critical parameters in our diffusion experiment are the fact that we did it at 150 micron isotropic resolution, and we implemented a long TE to maximally suppress any contribution of myelin. And then we fit the naughty model to this thing with parameters that were previously pseudo-validated by Wang et al. In 2019. And when we perform a similar analysis to the one with myelin water fraction imaging, we get very few statistically significant differences between the genotypes. And while our correlation studies show that these are somewhat correlated, their slopes are really shallow. So we can essentially say that within experimental error, that we have a relatively stable axonal density, and our signal is coming from specifically myelin and not from axonal loss in this very idiosyncratic hypomyelinating mouse model strain. So we then went and tried to look at light chain neurofilament as a surrogate for axonal density and repeated this experiment with LNF. And unlike the MBP and MWF experiment, we do not get clustering according to genotype. And both LNF and AWF from Naughty corroborate our hypothesis that we're mostly detecting a myelin signal here. So moving on, armed with those two pieces of information, we thought, hey, what the heck, why not? We might as well try to calculate the MRI G ratio. So we used a biophysical model previously developed by West et al. To scale our myelin water fractions to myelin volume fractions based on mass density arguments about myelin. And then we applied the Stickoff formalism to compute the aggregate bulk MR-G ratio within a voxel. And we got these interesting looking G ratio maps whereby we can see that as a function of mouse strain, we get progressive hypointensity in the G ratio maps, suggesting that in this implementation, the changes in G ratio are completely driven by loss of myelin water fraction and consequently by our MVP specifically. And finally, performing a similar regression analysis, we can see that we have a very high linear correlation between MVP mRNA and MRI G ratio. So that's like a intermediate step to validating G ratio measurements, although I'll say that very, very gingerly, and I'm sure some of you in the crowd will have a thing or two to say about that. Anyway, to summarize, we found this cool technique and we were implemented in Python, and we applied it to these interesting mice, and we found that rPCA-MWF scales linearly with MVP mRNA, and we validated this with MBP-IHC as well. And we found that rPCA naughty-derived aggregate MRI-G ratio is driven by reductions in MWF and therefore MVP. And currently we're working on some experiments to validate this with electron microscopy using scanning electron microscopy, and this is a current work in progress. So I'd like to thank everybody from the Shark Tank, from the BIC MR unit, as well as Dr. Alan Peterson and Human for developing the mouse, help from the GCRC for the histology, as well as funding support. Thank you very much.
Thanks a lot, Vlad. That was really good. A lot of work went into that. Is there any questions? Maybe one in the interest of time?
Yeah, there is one from Olivier Parent. What are the advantages of the PCA method to reconstruct MWF maps above the computational efficiency?
Sure, yeah. So basically, the non-negative least square technique, which is commonly applied to multi-echo spin echo, is ill-posed, and it takes forever to run, like we're talking about hours without parallelization. So people have generally applied a non-linear least square fitting to the sum of three exponentials for MWF calculations from gradient echo. And that is essentially really, really sensitive to it. First of all, you need this three-pool model, and then you need initialization parameters for the search algorithm to perform its gradient descent. So now the problem with that is that the manifold of the solution space is very wonky looking. So instead of trying to non-linearly fit this thing that's really sensitive to initial conditions, this employs absolutely no model whatsoever. It basically just performs a series of matrix calculations on the gradient echo data set. And there's a whole bunch of math that goes into it. So if you'd like to jot down this citation and check it out for yourself, I would love to discuss that in more detail, but it's like 3 pages of algebra. We managed to implement it and it seems to work, but the point is that model free, And because it's matrix calculations, it can run fast. So that's the short answer.
Great, thank you so much.